Okay, so this video might be a bit longer than usual, but I would like to go through the different steps of a process called cellular respiration. Cellular respiration is where we take glucose and oxygen and we're going to produce carbon dioxide, water, and energy. So we are going to break down this molecule of glucose, which is, as you remember, is a 6-carbon ring molecule. We're going to break it down through different processes all the way until we get the products carbon dioxide, water, and energy. Now, saying the products is energy is a bit misleading because it's not only energy. This energy is being used in order to create ATP and also heat, of course. So energy is going to give us ATP and heat. The heat is going to heat up the cell in our body, but the ATP is the most important thing. Now, remember this point. Each glucose molecule is going to yield through cellular respiration 38 ATPs. Now, in recent years, there is a lot of discussion about the amount of ATP that is being yielded from one glucose molecule, and it's mainly important to understand that the efficiency of the cell varies between and among different kind of cells. So it's not always going to be 38 ATP. Sometimes it's going to be 25, 35. On average, the number that we agreed on is one glucose molecule is probably going to give us around 39, 30 ATP. But for the IMAT and BMAT exam, remember that in total, the number of ATPs that can actually be produced from one glucose molecule from the entire metabolic pathways all the way to carbon dioxide, water, and energy, and ATP, is 38 ATP. And we are going to break it down by down in the next video, so don't worry about it. This video introductory might be a bit overwhelming for you guys, but bear with me, it's going to make sense in a few videos. So glucose enters the cell through a spatial pore that we are going to, a special protein that we are going to study on in the future. It enters the cell. When the glucose is inside the cell, we have a process called glycolysis. Glycolysis is the breakdown, the lysis of glucose molecules. And you are going to have a 10-step series, 10-step pathway that is going to take the glucose monomer, which is a 6-carbon sugar, and we are going to convert it through 10 steps into 2 molecules of something called pyruvate, which is each one is a 3-carbon sugar, 3-carbon molecule. Now, the thing is, many people ask me that we have 10 steps when we need to create 2 molecules that basically are the half of glucose. And if there are only 3 carbons, why do we need to have 10 steps in glycolysis? So we have 10 steps in glycolysis because between these 10 steps and along these 10 steps, first of all, we can also branch to other processes and metabolic pathways. And second of all, we are going to slowly yield the energy in an ordered manner in order to prevent chaos inside the cell, in order to encourage the organization and do everything in an organized manner inside the cell. To control the energy transfer between the substrate, it's much better to have multiple steps compared to one step. We can yield the energy and we can take the energy more efficiently when it comes to a multiple step, comparing to just one step. Also, of course, we have more metabolic pathways that are going to be connected to glycolysis. So we're going to make glycogen and we are going to do something called the pentose pathway. We're going to have many different pathways branching from this glycolysis. So it makes sense that this pathway is basically 10 steps and not a single step. So this is why we need to have multiple steps in metabolic pathways. Let's continue. So this glycolysis, which is the breakdown of sugar inside the cytoplasm, is going to give us 2 pyruvate molecules. The glycolysis is going to use 2 ATP molecules for 1 glucose molecule. So to derive the glycolysis, at least the first part of glycolysis, we are going to use 2 ATP, which doesn't really make sense because we want to make energy from these metabolic pathways. So we are going to use 2 ATP, but eventually all over the metabolic pathway we are going to also get 4 ATP for each glucose molecule. So we are going to get a 2 net of ATP. We are going to get 4 ATPs, but we are going to lose 2 ATPs. So the net income of the ATP is 2 ATP for each glucose monomer. And it's very important as well. So how many ATP do we get from ATP? We get 2 ATP for 1 glucose molecule for each time we do glycolysis. Now, this metabolic pathways is one of the most ancient metabolic pathways we have in nature. You can see it in prokaryotics and in eukaryotics as well. And it occurs inside the cytoplasm. It doesn't occur inside the mitochondria. So the next step would be to take our pyruvate that we just got. Let's say we have a eukaryotic cell, and we are going to get 2 pyruvates that are going to basically go to the mitochondria, inside the mitochondria. While they are going to the mitochondria and migrating to the mitochondria, they are going to have some chemical modifications, which is also going to yield some ATP, which we are going to study in the future. And the new metabolic pathway that we are going to have inside the mitochondria is actually called the Krebs Cycle. The Krebs Cycle is another metabolic pathway that is also going to yield some ATP and some more molecules that we are going to study. And it is also going to give us ATP. So for each cycle of the Krebs cycle, we are going to have 1 ATP. But because we have 2 pyruvate molecules from 1 each molecule, we are going to get 2 molecules of ATP. 2 ATPs in total for one glucose. Okay, I know it's complicated, so let's stop and analyze what we did so far. 1. Glucose enters the cell through a special protein. 2. Glucose goes through 10 steps that called glycolysis, which are basically going to consume 2 ATP, but eventually going to give us 4 ATP. And it is also going to be branched into different metabolic pathways. Glycolysis eventually is going to yield 2 3-carbon molecules, which are called pyruvate. One of the ways to use pyruvate depends on which type of cell and which are the environment you have in your surroundings, is to put them inside the mitochondria and to chemically modify them into a new molecule called acetyl-CoA, which we are going to study. And this acetyl-CoA is going to go through the cell cycle, inside the Krebs cycle, The Krebs cycle is basically yielding 1 molecule of ATP for 1 pyruvate, 1 acetyl-CoA, because acetyl-CoA, you will see, is the result of pyruvate through chemical modifications. This ATP is also going to be beneficial for the cell. So we have 2 ATP from the Krebs for 1 glucose molecule, and we have 2 ATP from the glycolysis for 1 glucose molecule. And now we are going to reach the highest yield and the most efficient way to create ATP inside the cell, which is basically inside the mitochondria. We will learn that on top of the inner membrane of the mitochondria, we have different structures. And these different structures are going to be loaded with electrons. The electrons are going to actually be brought to us from the glucose in the breakdown of the molecules. And you're going to see that we are going to use the electrons that we got from the original glucose and through the steps in order to yield a lot of ATP. And this process of creating ATP from the actual electron that we are going to get is called oxidative phosphorylation. I know it's complicated, don't worry. Phosphorylation. I mentioned electrons, which is a bit weird, and I know it's overwhelming because this is the first time we hear about electrons when it comes to energy and yielding inside a cell. Don't worry about it. Just know for now that during the steps of glycolysis and then the Krebs cycle, we have different molecules that called electron carriers. And during the steps of the glycolysis and Krebs cycle, the multiple step, they can just come and grab one of the electrons or two of the electrons that you have on the different substrates of the glycolysis and the Krebs cycle. So during the breakdown of the molecules, we can also lose electron to these carriers. And these carriers are basically going to go and to charge this new reaction over here. They are going to load the electrons right over the different proteins we have inside the mitochondria. And then they are going to go back and they are going to take another set of electrons. Then they are going to go back and charge it again. It's a cycle. You will see why it's so important and how electrons actually provide the energy to these proteins in the next few lessons. But for now, let's sum this video up because this is one of the longest video we did so far. And I know it's a bit complicated, so I'm going all the way again. I know it's very, very important topic for the IMAT exam and the BMAT. We have a cell, a eukaryotic cell. Glucose enters through a specific protein. We have glycolysis. Glycolysis is going to give us 2 ATP. Then we have the mitochondria, which is not the size of the mitochondria. The pyruvate, which is going to be the result of the glycolysis, is going to go inside the mitochondria, is going to have some chemical modifications that's going to yield some extra ATP, and it is going to go through inside the mitochondria through a process called the Krebs cycle. The Krebs cycle is going to also yield some ATP. Overall, during the glycolysis and the Krebs cycle, you have this enzyme, these coenzymes called FAD and NAD that we are going to study. And these molecules, these coenzymes that basically act as electron carrier, can come to the different parts, different steps between the glycolysis and Krebs cycle, and they can steal electrons from the substrates and the products of between the different reactions. They are getting charged with the electrons, they hold the electron, they go to the Krebs cycle and inside the matrix of the mitochondria, and they also take from the Krebs cycle and they go to the structures inside the mitochondria. They yield and they use the electrons, they drop them on these proteins that we are going to study on. They are going to derive the energy to do a lot of ATP, then they are going back to take some more electrons from these processes of glycolysis and the Krebs cycle. So we have this cycle of the coenzymes, the molecules that are basically electron carriers that are going to take the electrons from the different substrate and steps inside the glycolysis and the Krebs cycle, and they are going to charge different molecules that are found on top of the inner membrane of the mitochondria in order to provide the vast majority of ATP, which is around 34 ATP. So the vast majority of ATP is going to come from this process called oxidative phosphorylation. This process is called oxidative phosphorylation because we have to have oxygen in order to drive it. So we have glycolysis, then we have Krebs cycle, then we have oxidative phosphorylation. Glycolysis, Krebs cycle, oxidative phosphorylation. Glycolysis is an anaerobic process. It can occur without oxygen. However, the Krebs cycle and the oxidative phosphorylation, they must have oxygen. They are aerobic. But glycolysis is anaerobic. Because glycolysis is anaerobic, it actually allows many different organisms to yield glucose through a process that basically takes the pyruvate and creates another molecule that is not pyruvate, and we use it in different ways. So you will see that the glycolysis, the pyruvate will have many different fates other than the mitochondria. It will also have fermentation and lactic acid metabolism, but you will see it in future videos. I think I overwhelmed you enough with this video. So I'm not sure if this video made sense. It was a very, very broad introduction to what we are going to do and what we are going to study when it comes to actually cellular respiration and the yielding of glucose all the way to energy. In the next videos, we are going to break it part by part by part by part, and then you're going to actually understand what I was talking about these 15 minutes. I will see you next time.